Good morning. I'm John Morris, and I'm the director of the Memory and Aging Project and the Knight Alzheimer's Disease Research Center. On behalf of all of the faculty and staff of the Memory and Aging Project and the ADRC, we are just delighted to have all of you join us for our annual participants meeting. We have a great program, I believe, and full of interesting information. And as always, at the end of the program, we'll have all of the presenters come up for a question and answer period. I think it's been communicated to you that at your table should be cards and pens that you can write questions, and our team will come around and pick them up and bring them to me. We try to get through as many questions as we can, but we cannot guarantee that we'll get to all of them. So just a few housekeeping details. I think my first slide please take your cell phone or whatever you have and put it on mute or vibrate or something. Second, many of you have been here before, but not all. So if you have restroom needs, you go through the doors and turn left, just down that hallway. The refreshments, the breakfast and the coffee will stay out for some time, and you're free, of course, at any time to get up and refurnish your breakfast and your coffee. So I want to indicate that we began over 25 years ago the idea of a participants meeting to do two things. One, to thank all of you for your participation. Without you volunteering your time and effort with us, there would be no Memory and Aging Project. So we are here, all of us, to thank all of you. And second, we are here to relay to you the results of many of the studies in which you've participated. We're all united, all of us, in the effort to fight memory disorders, particularly Alzheimer's disease, and we want to keep you informed of our progress in that fight. Now, let me see. I don't really remember the slides I put together for this, so let's see what the next one is. Maybe it'll be a surprise. So here is our group, our faculty and our staff at the Memory and Aging Project and the Knight Alzheimer's Disease Research Center. And, you know, I'm biased, but I think it's the finest set of investigators working together on Alzheimer's disease anywhere. And also our staff is just terrific. Now, many of you, as you pulled up, saw signs and balloons and people standing outside and people assisting here. Our staff is also the finest of any staff in the memory and aging world. So let's give them a big thanks. And putting this participants' breakfast on takes the work of many, many people, but I want to acknowledge Angela Oliver in the back. She's waving her hand reluctantly. Put it up high, Angela. Thank you for coordinating everything. Now, once again, I'll probably miss some of our faculty and staff, but if I call your names, I'd like you to just stand up. For our faculty, Dr. Tammy Benzinger, who heads our imaging core. Tammy? David Kaufman, who's one of our investigators from St. Louis University. David, are you here? Okay. In the back, standing next to Angela, is Dr. David Carr. He's waving his hand. And just very close to David is Dr. Randy Bateman. He's waving his hand. And then Dr. Suzanne Schindler, Dr. Jason Hassenstab, and Dr. Kathy Rowe. Will you stand up, please? And who did I miss? Who did I forget? Oh, Dr. Ghoshal, please, Duper Ghoshal, please stand up. Thank you. It's terrible to have memory problems, isn't it? So when we took this photograph, I want to highlight that sitting next to me is a gentleman, Mr. Norman Say. Mr. Say was the inaugural chair of our African American Advisory Board, which has been a remarkable group of individuals. As a matter of fact, I'd like to have our African American Advisory Board members who are present to please stand and be recognized. And the reason I asked Norman to be the original chair of the board is because he is universally respected in the African American and indeed the St. Louis community for his work over many years to advance equality and to battle discrimination. You know that this is the 250th anniversary of the founding of St. Louis, and at the History Museum, they have a wonderful exhibit about 50 different people and events and so forth, and featured there is Norman Say. A little bit younger, but he's one of the 50 people recognized for his contributions to the history of St. Louis. Norman, thank you. And so when Dr. Holtzman and Dr. Rowe and Dr. Bateman and Dr. Ghoshal and all the others give presentations, we show a slide of the Knight Alzheimer's Disease Research Center, this slide, and we always include the gratitude we have to all of you and your families for all of the support you give us. So once again, thank you for that. Since we last met last June, we've added some new individuals. You see their names here. I'm not going to have them all stand and be recognized. You will be hearing from Dr. Danielle Cobbs in just a few minutes, but these are our new additions. And we've also had some people depart from the Memory and Aging Project, and here are their names. And one in particular retired last November, our senior nurse clinician, Mary Coates. She's continuing to work 3 half-days a week at the Memory and Aging Project, but there's Mary, and let's all give her a round of applause for all she's given. So let me tell you what we as a group are trying to accomplish with our research mission. We want to identify Alzheimer's disease when it first begins in the brain, but prior to its develop to such a point where it causes symptoms of Alzheimer's disease. That is, we want to detect it before there's any memory loss, any dementia, any problem. We call this silent phase preclinical Alzheimer's disease. And the reason we are so interested in this is we think that by the time symptoms have occurred, the brain changes have progressed pretty substantially. So we would like to identify the earliest silent preclinical stages to try to be able to intervene at that early silent stage in hopes of preventing dementia from occurring. This, of course, is not novel to Alzheimer's disease. Many chronic illnesses do best when we prevent them. For example, hardening of the arteries, the buildup of deposits in our blood vessels probably begins in our 30s and 40s, but it doesn't cause problems such as a heart attack or a stroke until the 50s or 60s. And we've had success in cardiovascular diseases by trying to reduce that silent buildup of hardening of the arteries. We try to reduce blood pressure, we try to control lipids, we try to control diabetes, and that has led to success in reducing the rate of heart attack and stroke. We want to take that same approach to Alzheimer's disease. So we want to learn, do people who have this preclinical stage of Alzheimer's disease, does that mean they always will develop the Alzheimer's dementia, the symptoms of Alzheimer's disease? Is it inevitable? We don't know the answer to that. Can we identify the stage where people with this preclinical stage, buildup of Alzheimer's changes in the brain, but still asymptomatic, is there a period where the changes inevitably cause symptomatic disease? Can we characterize those stages? And finally, we want to identify the timing of this preclinical stage to introduce drugs to try to prevent the onset of Alzheimer's symptoms. So we used to say, and many of you may have heard, that we can only identify the Alzheimer's changes in the brain in people who have died and come to autopsy, and we look under the microscope to try to find those changes. That's changed in the past 10 to 15 years. We can identify those changes, Alzheimer's changes in the brain in living people. And we do that primarily through imaging, but also what bathes the brain? It's that cerebral spinal fluid, right? You've heard about that? It bathes the brain. Matter of fact, the spinal fluid is manufactured in the brain. That's where it comes. And it protects the brain against injury. It also communicates with the spinal cord, which is an extension of the brain, and protects the spinal cord. And so we can examine the spinal fluid because it was manufactured in the brain and look for the abnormal Alzheimer's proteins. If they're in the brain, they're reflected in the spinal fluid. So looking at the spinal fluid has been enormously valuable in detecting people who may have this earliest asymptomatic stage of Alzheimer's disease. We don't offer a spinal tap to you simply because you enjoy it so much. We offer it because it is so precious to understand the course of the disease, how it builds up. We can track that now by examining the spinal fluid. Well, this is a slide that shows that in the symptomatic stage of Alzheimer's, there are many processes that go on in the brain. What we talk about when we say Alzheimer's disease, we get a picture of someone who has memory problems, maybe mild or moderate or even severe. But in that symptomatic stage, there are lots of things going on in the brain. And we've been giving drugs to people in the symptomatic stage to try to address one or more of these pathologies. All those attempts to treat this with those drugs have failed. Probably because we're only addressing one of those aspects and not all of them. So we may need to consider in this symptomatic stage, if we're going to have effective therapies, combinations of drugs that attack each of these different facets. But that may not be the case in this preclinical stage. And notice this preclinical stage really is the majority of the time we have Alzheimer's disease. The symptomatic stage is sort of the end stage of this process. And maybe in this preclinical stage, because now we have these biomarkers, and look, the very first thing that goes wrong is a change in a protein called amyloid beta that we detect in the spinal fluid. That's the first indication there's something wrong. But we also can do other biomarkers and imaging tests, and we want to know this process. When do all these changes build up in the brain and cause the brain to fail and then the dementia to appear? We're interested in that transition phase because we want to treat people to prevent them from progressing to symptomatic Alzheimer's disease. So that's our major theme in the Alzheimer's Center. We have many other themes. You're going to hear about some of them today, but I wanted you to know that we have been really at the forefront of this effort to characterize Alzheimer's disease prior to its symptomatic stage and to try to initiate therapies in that initial stage when people are still asymptomatic, with the goal of preventing or at least delaying the onset of symptomatic disease. And I'm very proud to say that the very first trial of a disease-modifying drug given to asymptomatic individuals who are at very high risk of becoming symptomatic began at Washington University in December of 2012, the first ever in the world. It's in a very special group of people who are at very high genetic risk of developing Alzheimer's disease, and that trial is in something called the Dominantly Inherited Alzheimer Network, and the trial is led by Randy Bateman. Randy, first ever trial to prevent Alzheimer's disease. Thank you and your team for doing that. So this is you. This is some statistics on you, the memory and aging participants. You see it's almost 900 individuals. We have different grant mechanisms. That's what those abbreviations, HASD and ADRC, that support them. And the DIAN grant, the Dominantly Inherited Alzheimer Network that I just mentioned, is an international study. It's headquartered at Washington University, and the Washington University site has by far the most individuals enrolled in DIAN. Look at the age range that we have in the Alzheimer's Disease Research Center. Really, we're looking at the lifespan. The reason is we want to know when those brain changes first begin. And if we only look at people 65 and older, we're going to not see if it happens at 60 or 50 or 40. So we're looking very much at people throughout the entire lifespan, adult lifespan. 57% of you are women. I don't mean personally, but as a group. And 15% outside of the DIAN study are African American, thanks to our efforts of our African American Advisory Board. We've been very busy. We've been doing lots. 900 people seeing them annually is a big deal. One of the things that we just reviewed, and I was surprised and not very happy, is that in 2014 we wanted to recruit a certain number of new participants. So we wanted people who don't have dementia, cognitively normal. We wanted to enroll 38 over the calendar year 2014, and to date we only have 8. People with symptomatic Alzheimer's disease, dementia, we want 27. We're a little better there, we're about 12. And of all of these individuals, we want at least 20 of them to be African American. We've only enrolled 6. So I'm going to ask all of you to help us, if you know of people who might be willing to be a participant, please encourage them to call us. There's our phone number, 286-2683, for further information. We're looking primarily for people 65 and older who are in good health. We'll have someone to accompany them to our assessments, a study partner, and don't have any contraindications to participating in our imaging and spinal tap program. We particularly need cognitively normal men. We have about 67%, almost 70% women, and we want to have it balanced. So I'm asking all of you for help. Our phone number is there. Terry Hosto, where are you, Terry? And Becky Fearberg are standing up. Are they? Becky is in the back. Okay, she's raising her hand. And where's Terry? And Terry's up here. So we've got the room covered. But if you know of people and you want to communicate to Becky and Terry their names and information, and certainly encourage them to call and volunteer. An error on the printouts. Okay, I'm told that the printout doesn't have the accurate phone number, so please, let's write it down, make sure you have it. Area code is 314. Then it's 286-2683. No, okay? I get really confused. I remember, Mary Coates may remember this too. Once I was up here telling you to call our phone number, and I was giving you the wrong phone number, and Mary kept trying to come up and tell me the right phone number, and I didn't want to listen to her, but that's the right, 286-2683 is the right phone number. So I mentioned that we're one of the, in my opinion, top Alzheimer programs worldwide, and we've been very fortunate to get funding from primarily the National Institutes of Health. And for many years, we've been the top funded Alzheimer's disease research program. And this year, we're number 2. A group in Arizona has been funded after the Diane study started. They also launched a secondary prevention trial. And they're studying it in South America, in Colombia. And they have some major NIH support to do that, but we're still very highly funded. These are our 4 different grants. The Alzheimer's Disease Research Center, the Healthy Aging and Senile Dementia, the Adult Children Study, and then the Dominantly Inherited Alzheimer Network. I include this here just to give you an idea of the kinds of science projects that goes on in these. So we're looking at imaging for different proteins, and we'll correlate them with CSF biomarkers. CSF means spinal fluid. We're looking how that abnormal protein I mentioned, A beta, gets cleared out from the brain. We're looking at whether our diurnal patterns, including sleep, may have something to do with this. We want to identify the transition from asymptomatic to symptomatic. We're looking at a whole host of sleep parameters, and we're trying to look at the genetic risk that people have. So these are just some of the science projects that we're doing in a coordinated way across the entire ADRC. I mentioned our funding comes from the National Institutes of Health. Every 5 years or so, we have to reapply to continue that funding. We've been very fortunate. As I said, we're one of the top funded Alzheimer's centers. But our big grant to fund the ADRC just went in about a month ago. It was a huge grant, almost 1,000 pages. And we're requesting about $14 million over the 5 years to do the study. We think we merit this because we've been very productive in the last 5 years, the current funding period, over 600 scientific publications. We've supported over 280 studies that have used the ADRC to go on and themselves get funded, $158 million based upon what we're doing in the Alzheimer's Disease Research Center. And the very bottom one, I want you to look, if you total up all of the new funded research that was leveraged by the ADRC, we have almost $200 million that has been supported by the ADRC, and our current budget period only about $10.5 million. So we think that's a pretty good return on the NIH's investment in us. Here are just some of the studies that we are doing. You're going to hear about a few of them today. We do a lot of imaging studies, and it's led by a number of individuals. You can see their names. We look at fluids, both blood and spinal fluids, and you can see the kinds of testing we're doing there. We're doing genetic studies. We test, of course, memory and thinking, cognition, and a number of other studies in sleep, in vision, and in driving. Now, some of the coordinators for these studies are here, and I'll ask them to stand. So is Anna Kim here? Anna Kim is at the front of the room. So she's coordinating a study by one of our imaging individuals in the Department of Psychology, Denise Head, to look at the relationship of stress and brain structure and cognition. So if any of you ever get stressed, this is a great study for you. And afterwards, you can talk with Anna to find out more about how to participate. David Kaufman and Ryan Van Patten, are they? Okay, good, in the back of the room, good. David is here. He's interested in brain activity, measuring it through picking up the electrical signals of the brain, something called electroencephalography, or EEG, and he wants to have people do cognitive tasks and see how that may or may not change the electrical activity. Jennifer McLeland is here. Jennifer is standing. She's way in the back. Stand and wave. People are still turning around. There you go. And she's helping in a number of studies doing related to sleep and the possible role of sleep in Alzheimer's disease that you'll hear about from Dr. Holtzman. So if you're interested in that, please speak with Jennifer. Kara Ramsey, is Kara here? Yep, and she's standing in the back too. Oh, the back's popular. She's a second-year medical student, and she's working with Randy Bateman to try to find out biomarkers in the cerebral spinal fluid, and also to see if we can develop a blood test for Alzheimer's disease. I know all of you would hate to give up the spinal fluid test, but say we got a blood test, that might be good. And then Kathy Rowe was introduced earlier. Kathy stands up. She's doing the driving study in healthy people to see how whether preclinical Alzheimer's disease affects that. Some of you have been notified that we had a clinic at Barnes Jewish West County here on Olive Road west of 270. We started that clinic when the Highway 40, Interstate 64 construction or reconstruction was going on, because we thought maybe many of our West County participants wouldn't be able to negotiate the construction. So we started the West County program at that time. Even when the construction was completed, it was so popular that we've continued it. But I have to say, now with the workload that we have, our 900 annual participants, trying to staff 2 clinics has become impossible. So with regret, we're having to close the West County clinic as of Memory and Aging Project clinic as of July 1. and people will all come to our home clinic, the Alzheimer's Disease Research Center at Washington University School of Medicine. I wish we could continue it, and if we have some more funding, we'll try to restore it, but at present we're closing the West County office. All right, well, enough of me for now. You'll get me later. This is what we're going to be discussing. Updates to new initiatives within our program. So I want you to hear about a satellite for our Memory and Aging project. And Dr. David Carr will, David, are you going to introduce? You'll come up and introduce. And then a new program I mentioned, Kara is a second-year medical student for introducing medical students to individuals who have Alzheimer's disease. You know, the first 2 years of medical school are primarily in the classroom and in the laboratory and very little interaction with patients, and we wanted to try to expose interested medical students in those early years to patients and also to help them understand that people with Alzheimer's disease still have much to offer and to try to remove any stigma that people might have about that. So why don't we have Dave Carr come up and he'll introduce the Memory and Aging Project Satellite. David?
Thanks, John. I'm really excited to introduce our next speaker. I do want to just give a little nod real quick to the Memory and Aging Project Satellite Core. Especially the African American Outreach Satellite. Marie Meisel, could you stand up for just a second? Marie's been a nurse for, what, 25 years. And the goal of our satellite is to provide both in-home dementia assessments and also to help with recruitment, minority recruitment, in the Memory and Aging Project. So I would like to introduce Dr. Danielle Cobbs, occupational therapy. I think she just finished her doctorate. We're really excited about adding her to the mix. Danielle, come on up.
Dave,
for home assessment.
You want to introduce James?
I didn't know he was here.
Yeah, he's here.
Oh, before I do that, thanks. James Williams, if you could stand up. Are you here?
Yeah, he is.
Thank you. Social worker and Memory and Aging Project Corps. Also, over 25 years of service. Thank you, James. Very good.
Hello, everyone. Once again, I'm Dr. Danielle Cobbs, and I'll be informing you about our Memory and Aging Project Satellite. So who are we? We are, first off, an extension of Washington University's Memory and Aging Project. We're partners with the St. Louis Area Agency on Aging, and also we are a team of health and social service professionals working together to link older adults living in the community with memory loss with helpful resources. In turn, they participate in our research studies so that we can all learn more about memory loss related to Alzheimer's disease. Our team consists of, as Dr. Carr just simply introduced us, he is our team leader. We have Marie Meisel, who is our nurse coordinator. She's the one that administers the memory and thinking tests. She interviews the participants and their collateral sources or their informants, and she educates on memory loss. James Williams is the social worker who identifies needs for the clients and determines eligibility for supportive services. I, once again, am the new team member, the add-on to the team, the occupational therapist, and I'll be conducting home safety assessments and making recommendations. And then last but not least, we have Laura Ahrens, who is our research assistant that manages all of the participant information and keeps it private. We all work together to provide supportive assistance to promote successful aging in place for our participants. How we do this? It starts with the in-home assessments, and then we learn about the participants and their resources, and we'll be doing the new home safety assessments. This is all in efforts to assist with African American recruitment with the Memory and Aging Project, with other Alzheimer's Disease Research Center satellites, or cores, and then also with the community partner recruitments. Our intentions are to build rapport and also maintain relationships with our participants. Our services consist of the in-home memory assessment, education and counseling, recommendations identifying needs, and then also the home safety and community mobility participation evaluation, which I'll explain further shortly. To be eligible for our services, you need to be 60 years or older, living in the community with memory loss, and you need to have an active caregiver or a collateral source, someone who just knows you really well. So the new piece to our MAPS puzzle is assessing home safety and in the future community mobility. Why do this? Well, may I have all of our participants look to your left, the person on your left, and then look to your right. Why am I having you do this? I just want you to see each other. Everyone looks nice this morning. Well, according to statistics, 3 in 10 of you will have or have had a fall this year. Why is this? Well, people live in what we like to call Peter Pan housing. We move in thinking that we'll never ever grow old. But reality is, successful aging is often threatened by a number of environmental barriers that present in the home of older adults. So when we go into homes, we typically see people doing things like this. And although it's unsafe, they don't see it as unsafe. They see it as a way that they compensate so that they're still able to do the things that they need and want to do around their homes. Even when older adults don't identify that they have a problem within their home, they tend to have at least 4 environmental barriers that limit their participation in their daily activities. These barriers begin to happen as a result of just experiencing the normal aging processes that we have. As we begin to age, our personal abilities change, and that creates a mismatch between the home environment and our newfound abilities. This can also carry over into the community, which affects community mobility. So doing things such as walking around your community and driving become big problems. So that's something we want to look at in the future, assessing community mobility and community participation. Because when you're unable to get out into your community, you're not able to get to the doctor to get your medicines, you're not able to go shopping, or do the fun, productive activities we love to do, like bingo. We have 3 particular steps that we like to use in assessing the home environment with our fall assessments and with our home assessments. It starts with a home safety checklist and also evaluation so that we can identify the problem areas and address those areas. Then we assess fall risks and we develop personal prescriptions for everyone. There are several different solutions to reducing fall risks that include balance and strength training, Tai Chi, checking glasses, and medications. However, our focus with our recommendations will be on home modifications. Home modifications are just adaptations that we make to the home so that we're able to perform our daily activities as independently and as safely as possible. They can consist of adaptive equipment like bath benches, architectural modifications like our grab bars, task modifications, so changing the way that we do things, personal assistance, having someone else help us do things, patient and caregiver education, so teaching people how to properly do things, and then also referrals to appropriate services. Why home modifications? Why is this our emphasis? Well, home modifications are proven to help people function more independently and more safely in their homes. Furthermore, when home modifications are tailored to a person's specific wants and needs, people are more likely to use the things that we give or use the recommendations that we give. They're more likely to do things the way that we suggest that they do them, and fall risks are reduced. And so that is why we have added home safety and personalized prescriptions with tailored modifications to our MAPS puzzle. That's all I have, and I would like to thank you all for your time and participation.
Thanks very much, Danielle. We have part of our Alzheimer's Disease Research Center includes something we call the Education Core, which it translates the research that we do to the community. We just happen to have, not that I'm biased, the best Education Core. It's led by Andre Denny and Jennifer Phillips and Murtis Spencer. So they're all terrific, and Jennifer's here to talk about this dementia understanding opportunity. Please welcome Ms. Phillips.
Good morning. Thank you so much to everybody for being here
Hold
today.
the microphone closer to you.
Thanks to everybody for being here today, and it's my pleasure to be here and tell you a little bit about the DUO program, or the Dementia Understanding Opportunity Program, that is becoming one of my favorite parts of being on the team at the Knight Alzheimer's Disease Research Center. The idea behind the program is to match first and second year Washington University medical students with an individual with early stage memory loss caused by Alzheimer's disease. The pairs will meet together for 2 hours per month throughout the duration of the academic year, with an additional 1 hour asked of each student per month, where they gather for supplemental programming specific to Alzheimer's disease, related neurology topics, gerontology, or other topics that the students identify that are of interest. Through the program, we also request that care partners be involved as well. It's not required, but it certainly adds a rich depth to the program whenever they are able to participate. We have a lot of goals for this program. Although it's proven over time to be a very enjoyable social activity, we also have some outcomes that we're looking to achieve through the student and mentor participation. The first is that for the students, we're really looking to elevate their awareness of the skills and strengths that remain with individuals with Alzheimer's disease. We really want them to see firsthand that there's a very significant and a very high quality of life that is maintained long into this disease process. We do like to provide the opportunity to see how somebody responds to their own changing abilities, learn effective ways of communicating with individuals with dementia, something that will definitely translate into their practice over time, and also increase knowledge and empathy regarding aging and dementia with the added goal of reducing stigma. But we also have goals for our individuals with memory loss, our mentors as we call them. First, we like the idea of providing them with an empowering and an exciting way to remain active and stay involved in their community. So they have the opportunity to share their personal experience and their life story with an up-and-coming physician, and also not just get to know that student and coming physician, but really have the potential to influence their future as a physician. Thus far, the DUO program is in its second year, and the first year we matched 7 individual students with mentors, and the second year we've matched 10 students with mentors. And we found 10 to be a very nice quantity. It allows for good group interaction, and so we're going to stay with the goal of 10 people as we advance this program again into the third year. So we do have a selection process for identifying our students and also identifying our mentors. So when it comes to the students, the first thing to know is that DUO is an extracurricular activity, meaning that in the middle of a very busy first year of medical school or an even busier second year of medical school, these students participate in the DUO program because they're so interested in learning the kind of content that they won't receive in a classroom. So they complete an application and interview process to be a part of the program. And they also undergo some training. So they have 3 hours of initial training about Alzheimer's disease, behaviors, and communications. And then they also, as mentioned, have that supplemental 1 hour of training each month. Our diagnosed individuals join us from a variety of different partnering referral sources. So some of our partners might come to, our mentors might come to us from the Memory and Aging Project. They might join us from the Memory and Diagnostics Center, or sometimes we receive great referrals from our partners at the Alzheimer's Association, St. Louis chapter. So we also do a brief interview with our mentors as well. We want to make sure that they're able to articulate their experience with memory loss and share those feelings about the diagnosis. And we want to know that they're going to be able to participate in the program in a way that will ultimately provide content and value for both the mentor and also the students. Typically, we're looking for individuals that are in the earlier stages of the disease, but that's a very broad terminology. So we've had some people that are very early in their experience and can very accurately articulate what those changes are like. And we have some people that are a little bit further along in the process, and some of their experiences are explained a little bit more intangibly, and the student can see those through observing and interacting, and also by speaking with their care partners. And then ideally, as mentioned, the support of that care partner is so valuable to the program because that's an additional very rich relationship that can be developed, again, that is not accessible in the classroom. So in addition to all of those different outcomes, we also have some specific categories that we're capturing information in regards to. So we're also identifying increases in empathy, knowledge, reducing stigma, and then also measuring changes in students' attitudes regarding a diagnosis of dementia. Additionally, we're capturing gains in knowledge in a variety of different categories. And over the first year of the program, we've actually seen significant growth in the variety of different areas reflected on this bar chart. So for example, 80% of our students have increased knowledge in the symptoms of a memory disorder. And even something a little bit less tangible, but even 30% of students the first year experienced increased knowledge in aggressive behaviors just by virtue of being involved in the program. Not through anything that they experienced personally, but from hearing stories and interacting with the care partners and individuals, they've really been able to glean a sense of what the day-to-day life is like for somebody experiencing changes. The student feedback that we've received from our program has been incredible. This particular bit of feedback I have is shared from our partner site at Northwestern University, which was actually the incepting site of the current version of the DUO program that we know today. They have a program called the Buddy program, which has been replicated by Dartmouth University as the Paris program, or pardon me, Boston University as the Paris program, and also a fourth program at Dartmouth University. But students have indicated not just a growth in knowledge, but a growth in their capacity for empathy and compassion for persons with dementia, an increased understanding of the changes people are experiencing, not just textbook comprehension, but actual real-life implications of what these changes mean. And finally, an appreciation for the challenges for the diagnosed individuals and their families. And I think we can all agree that these are gains that will be very beneficial to a physician, no matter their specialty, as they go out and start their practices. So I can tell you a lot about the program and what I believe is such a wonderful and valuable experience, but I'm very pleased today to have some individual participants who have been with us in the DUO program this past year. So at this time, I'm very honored to invite Morcy and Janice Cole up to the podium to share their experience, Morcy as an individual mentor in the program, and Janice as his care partner.
Good morning, everyone. My name is Morcy Cole.
Get a little closer to the microphone.
Yes, again, for the people who haven't heard, my name is Morcy Cole. I'm a novelist, person who don't know too much about this. Okay, I'm sorry. You want to start first? Okay. All right. I was going to go one way, but now I got to go another. Just to tell. Jordan, which is a friend of mine, I think it was about at least eight months ago when my friend here now is Jordan, and in school and stuff like that, and trying to do as much as he can. And he's trying to make me, you know, somewhat interested in it, you know. I'm, again, just a novice on it, so what I'm going to do is to give it to my wife so she can end it. Literally.
Hello, everyone. I'm Janice Cole, and I am here to help Morsi to explain how he enjoyed being in this duo program with Jordan Stanley. When we first started out, we didn't have a lot to say, and Morsi was kind of nervous about going to, we mainly went to a restaurant to eat. And I was wondering, you know, how it was going to work out, but each time that we went, those 2 hours, Morsi was always wondering about, what are we going to say? How long is, you know, that's a long time. But then the 2 hours would pass, and he would say, is it time to go already? So the thing that I liked about it, and I think Morsi can attest to this too, is that Jordan was a very compassionate student who really took so much time and patience with Morsi and listened so much to what he had to say. In fact, Morsi spoke more about his background and his history than he has talked with me in the last 38 years. And it's like I'm sitting there saying, oh, really? You know? So some of the stuff was new to me. So he brought out, I can see how the program is very beneficial, because the person with Alzheimer's and the person they're with, they're sort of strangers, but they don't mind, you know, getting together and saying things that maybe they just hadn't had the opportunity to say, or at the time that it came up, it just came out, and it was perfect. I enjoyed Jordan quite a bit myself, and I just hope that the things that we were able to say and do during the time that this program is helpful to him. I feel like, you know, if when I go to the doctor, and me and Morsi, I'll take Morsi to the doctor, now the doctor that we go to now probably didn't have this program, but I can see in the future for doctors who will have this program and have an opportunity to sit and talk to people, that not as a patient, but as a person to person, that it's going to really benefit you in your practice later. Thank you.
Thank you, Morsi, and thank you, Janice. And I'm also pleased to invite Jordan up to join us as well. Jordan just wrapped up his first year of medical school and is also with us as a student this summer. So we're pleased to have Jordan.
Thank you, everyone. So yes, I just finished my first year of medical school at Washington University, and I had the distinct pleasure of being in the duo program this last year, and the honor of calling Morsi and Janice my friends. So the reason I wanted to join the duo program was because I'd already had some background in psychology and neuroscience, and then I was getting a lot of that in medical school, trust me. But what we were learning about was the biochemistry of the disease, we were learning about the genetics of the disease, we were learning about the physiology of the disease, but we weren't talking about the personal aspects, and I was really interested in that. And I got the opportunity to learn them from my time in the duo program. So we would get to talk about the logistics of going out in the community and staying active, and how do you do that if you're not driving or if you don't have as many contacts as you used to. And we would also talk about the financial implications and the emotional implications, and it was very enlightening and a great way to complete the circle and not just hear about the medical aspect, but hear about the personal aspect. And I know that it's really helped me so that when I'm later a physician, I'll be able to see patients not just as a disease, but as a person and as a life story. So I found it very valuable. My favorite experience was they recommended that I come with them to their Alzheimer's Support Center. They had Project Esteem, and they spoke so highly of this program, they recommended that I come see it. and it was a great pleasure to be there and see what a warm, supportive environment it was and what a great community they had established. And I very much enjoyed myself. So thank you, and thank you, Morsi and Janice. It was a wonderful opportunity.
Are you finished?
So we just want to conclude with saying that we're very thrilled to be entering year 3 and continue our collaborations with the other partner universities. We are always interested in talking with individuals that would like to learn more about the DUO program. So we are here. I will be at the table in the back. Our Education Corps leader, Andre Denny, is also going to be back there if anybody has any interest in learning more. Or my name and phone number is there, and we welcome you to call us at any time.
So thanks, Jennifer. It's a unique program and very innovative. Jordan mentioned Project Esteem. I want to indicate that that is a program for our St. Louis chapter of the Alzheimer's Association, another great partner of the Knight ADRC, and the president of the St. Louis chapter, Stacy Tu-Lavaz, is with us today. Stacy, will you stand up? Thank you very much. So really interesting connection between Alzheimer's disease and sleep is emerging, and one of the leading investigators in this is Dr. David Holtzman. He's the Jones Professor and Chair of the Department of Neurology at Washington University and Associate Director of the Alzheimer's Disease Research Center. So please welcome Dr. Holtzman.
Good morning, everybody. It's really a pleasure to be here, and I look forward to telling you about a new study that we're starting on sleep. Before I do that, I wanted to thank all of you, as Dr. Morris did, for being participants. I have the great pleasure and honor to be able to travel throughout the United States, but even throughout the world, and one of the things that's very striking, whether I have visited either Japan or Australia or Europe, is that when I give talks about my own research or the research that I'm doing that's related or with the Alzheimer's Disease Research Center, the work that's going on here is so well known, they know all about you all. And the memory and aging participants are really world famous now. So it's all of what you're doing has really contributed in a major way to our understanding about this disease, and I'm sure it will lead to treatments in the future. So thanks for doing that. So what I'm going to tell you about are studies that we are doing to look at the sleep-wake cycle as a potential marker for what John referred to as preclinical Alzheimer's disease. And these studies, while I'm leading them, they're being carried out by Brendan Lucy, one of the neurologists that's in the Sleep Center, Kel Yamada, also a professor of neurology who leads our Sleep Center, and Ann Fagan, who runs our biomarker research core. So just to give you some background, sleep is very important, as you probably all know, for your normal health, including for memory and thinking. If you think about it, when you have a good night's sleep, you wake up, you think clearer, and if you don't, usually it's kind of hard to think as clearly. Many people who develop dementia from Alzheimer's disease also, as you probably have seen if you have personal experience, have sleep problems. We have actually been studying not only humans, beings who have Alzheimer's disease or may develop it, but we've been studying special kinds of mice that develop changes of Alzheimer's disease in their brain. And we have two major findings I will tell you about that have led us to want to do further studies. One is that if we sleep deprive mice, we prevent them from sleeping as much for as little as a month or two, they develop more Alzheimer's changes in their brain, a lot more actually. And the other thing that we find is that once the mice develop these Alzheimer's changes, their sleep deteriorates. So I'll show you a picture of this. If you look on the panels where it says on the left, control, these are brain sections from a mouse that is living its life, and it's predisposed to get Alzheimer's changes. And what you can see are there's a few little dots, these little brown dots, and those dots represent amyloid plaques that build up in the brain in Alzheimer's disease. And you can see there's just a few of them on this picture. But what you can also see is that if you look on the right, these are mice that were sleep deprived for just 4 weeks. And they developed about 3 times more amyloid plaques in their brain. Now, a mouse lives a lot less time than a human, only 2 years. So sleep depriving mice for a month is equivalent to sleep depriving humans for years. On the other hand, it's a pretty substantial change, and I think that's something worth further investigating. The other thing that we looked at is we looked at these mice when they were 3 months old, 6 months old, or 9 months old. And what's shown in this graph is the amount of time the mice are awake when they should be sleeping. And what you can see is that when they're 3 months old, they're awake a certain amount of time, say 20 minutes per hour, when they should be sleeping. That's normal for a mouse. But the 9-month-old mice are awake more than half the time when they should be sleeping. And what we found is that the buildup of this amyloid protein is leading to that problem in sleep. Okay? So what we have done so far already to address this, I think you may have heard from, if you came in the last few years, from Yoel Ju, one of our other assistant professors who's in the sleep center. And what she did is that we carried out a study in participants in the adult children study. I know some of you in the audience are participants in that study. And she used a technique called actigraphy. This is a wristwatch device that measures movement, and you just wear it like a watch. And what we found is that sleep efficiency, so this is the amount of time that you're sleeping while you're actually in bed. So if you have efficient sleep, when you get in bed, you sleep most of the time that you're in bed. If you don't, you could be awake a fair amount of time in bed. So what we found is that sleep efficiency is decreased in people who have amyloid deposition. These are still people that are otherwise normal. Neither we nor the participants we studied could tell they had any problems. They weren't reporting they had problems with sleep. We only could pick this up with the device. So this suggests that we might be able to detect sleep changes using more sophisticated and accurate testing methods in people in the Memory and Aging Project. So if that's the case, this would allow us to detect Alzheimer's disease before the changes in memory and thinking occur, and what we hope is that we could potentially monitor the effect of new treatments to delay the disease using this relatively simple technique. But we can't do that until we know that it works properly. So how do we do this? So we have a new study that we'll be telling some of you about and asking you to participate in using a device called the Sleep Profiler. And you can see a picture of a woman wearing the Sleep Profiler. It's basically just like a headband that you wear around your head. I tested it and wore it several nights last year, and it didn't seem to bother me too much. It's fine. It's just like wearing a band. You go to sleep with it. And what we're asking people to do is to wear this for 6 nights, basically, where they would wear one device for 3 nights, and then we give them 2 devices, and they wear the other device for 3 nights, and then they simply return the device to us, and we analyze the information. So you'll hear about this more when you come to the Memory and Aging Project to participate in the other things that you're doing. I want to show you just the kind of information we get from the device. So what you can see is the device essentially does what's called an EEG, an electroencephalogram. It measures your brain waves. So what you can see on the top, if you look closely, you see all these little squiggles. This is measuring every second or 2 brain waves. And you can see when somebody's awake, you see these relatively frequent waves that go up and down. What you can see when somebody's sleeping is that the waves are much less frequent. They're bigger, and they're less, the peaks and valleys are wider. And that allows us to say how deep somebody's sleep is. light sleep, moderate sleep, or deep sleep. What we think is that the better deep sleep you get, that might be protective, and also might, if it gets less, that might mean that you're developing some of the Alzheimer's changes. That's what we're testing. So the things that we measure are wakefulness, what we call non-REM, or the deeper forms of sleep. There's different stages of the sleep, N1, N2, N3. And then we measure REM, or rapid eye movement sleep. That's the sleep that occurs when you're dreaming. So we really look forward to all of you that get asked to hopefully participate in the study. We think we'll learn a lot from this, and hopefully it will help the field if we figure out some new things about sleep. So Jennifer McClelland, who Dr. Morris introduced earlier, Jennifer's in the back, I think, right? So Jennifer's one of the Sleep Center technicians, and she's here with us, and she can answer any specific questions about the study after today's event. So thanks very much for being here, and we'll take questions at the end.
So thanks, Dave. We ask you participants to undergo a number of our imaging studies, and you know that you have to generally lie down on a table for a period of time, and what actually is being collected? What are we studying? So Dr. Manu Goyal, who has been trained both as a neurologist and just completing training as a neuroradiologist, is going to explain what goes on when you're in the scanner. Manu?
Well, good morning, everyone. It's truly a privilege to be here and to be able to talk to you about some of the things that we've been learning with radiology and imaging of Alzheimer's disease. I should note that much of this is led by Tammy Benziger, who leads the imaging core. It's been very helpful in putting together these slides. In understanding the human brain and Alzheimer's disease is very challenging, of course. And we have, over the last 10 to 15 years, developed many tools to better understand the brain. So for example, we have a very advanced form of MRI that lets us look at the structure of the brain as it ages, as it starts developing memory problems. We can use the same MRI machine with new tricks to look at the function of the brain, to look at how different parts of the brain are talking to one another. We also have a PET scanner, and some of you may know that PET imaging was largely invented at Washington University. We can use this to look at how different parts of the brain are using energy. And then we can also use the same PET scanner to look at different pathology in the brain. Amyloid is one major culprit in Alzheimer's disease. We can use a PET scanner to look at amyloid in the brain. And the other culprit in Alzheimer's disease is tau. And we're excited to say that now we can use PET imaging again to look at tau in the brain. And what's particularly important at Washington University is that we can do all of this at the same time. So we can look at many different features of the brain and integrate all of this, not just imaging, but also with spinal fluid analysis and genetics and other studies. So let me give you one example of what we've learned in the past few years. You might have heard of a myth that we only use 10% of our brains. In fact, even at rest, our brain is very active. There are many regions in our brain that are active at rest, and our own Professor Marcus Rakel has identified those regions and called it the default mode network. Well, it turns out that we can see the default mode network using MRI, and that's shown right here, as well as PET. And what we find is that those same regions develop amyloid in Alzheimer's disease, which is shown here. And in Alzheimer's disease, the regions that lose their ability to take in energy are, again, very much the same regions of the default mode network. So by integrating MRI with PET, we're able to learn more about what is happening in Alzheimer's disease. we can also integrate this information across time. And so you've heard that the processes that lead to Alzheimer's disease begin much earlier, much before the symptoms begin, and we can see that with spinal fluid analysis as well as amyloid imaging. We can see how functional MRI determines how the different brain regions talk to one another, how they change as the symptoms of Alzheimer's disease begin to take shape. And then we can also see how the structure of the brain changes. So we think it's so important to integrate this information together that we are one of a handful of centers to own a PET MRI machine. We can perform MRI and PET at the same time. And this is very convenient to us because this is an example of a subject who has had amyloid PET imaging, had a brain MRI, all at the exact same time. And so this lets us put this information together very nicely. The other nice thing about having a combined PET MRI scanner is that it's much more convenient for all of you. So now we can perform 3 different exams, PET scans and MRI, in one simultaneous scan that can take about 1 hour. Previously, you would have to go to an MRI scanner first and then a PET scanner. Now we can do that all at once. And we also have a shorter exam for subjects who can't tolerate the full 1 hour. That takes about 20 minutes. One thing I want to note is that we also have a new way of imaging amyloid with an agent called fluorobetapir. Some of you might be familiar with PIB, which was one way of imaging amyloid using the PET scanner. Fluorobetapir has certain advantages, and it's something that can be used by other centers that may not be as sophisticated as our own center at Washington University, and it turns out that it's about just as good as PIB at looking at amyloid. So if you participate and you get a PET scan, some of you might be getting injections of fluorobetapir rather than PIB, and I should note that this graph here that has demonstrated this is in large part due to your help and in participation in this project. I'm excited to say that we can also now image tau in the human brain, and we're beginning studies to look at how tau might also be contributing to Alzheimer's disease. So I just want to provide some basic information of what to expect when you come to get imaged by MRI and PET. MRI is a very loud machine, so you'll be given ear plugs and headphones to try to mitigate that. It's a flat, hard surface. We would ask that you wear very comfortable clothing when you come to MRI. And also, you cannot wear any jewelry in the MRI scanner because it's metallic and this is a very powerful magnet. Finally, we will ask you to be very well rested because many of the studies require you to be awake. And believe it or not, it's easy to fall asleep in an MRI scanner. For PET, the PET scanning portion, an IV will be placed to inject some of the tracers to be able to look at amyloid or tau, or to look at energy in the brain. There are some safety considerations. If you have a pacemaker, you cannot undergo an MRI scan. If you have any electronic implant that cannot be removed, you cannot undergo an MRI scan for these studies. And we do have a detailed screening questionnaire that we'll ask you to fill out before we even let you near an MRI scanner. PET scanning also has associated with it a small dose of radiation. That amount is about the same amount that you get every year just for being on Earth. And it's quite a bit less than what the government allows people like myself, a radiologist, to experience every year. That said, there is theoretically a tiny increased risk of cancer related to radiation, but it is very tiny. So for example, if you have a 50-year-old man His lifetime risk of developing cancer is 35.8% for the government. And after one PET scan, that goes up to 35.9%. So it's a very tiny increase, though it is theoretical, and it's an important increase to take note of. So I would like to end with this slide just to, again, emphasize that we can use imaging to look at many aspects of the brain, how it ages, how it develops Alzheimer's disease, and then we can relate this information to genetics, to spinal fluid analysis, to different brain cells, and of course, ultimately to the clinical aspects of Alzheimer's disease. Thank you very much.
I mentioned earlier that we have had discouraging results from trials of experimental agents to treat Alzheimer's disease. We have a few new trials that we're going to participate in. I'm going to ask Dr. Randy Bateman to come up and give us an update on trials of new medications. Randy? Let's welcome Randy Bateman.
Good morning. It's great to see you all here again. I don't know how many times this has been for me, but I look forward to this each time I'm asked to come and talk. And this time I was asked not to talk about necessarily the research that we do in my lab, but to talk about the clinical trials, which so many of you have participated in. So I received a text this morning, as I often do, from my dad. He texts me almost every day. And he doesn't text me about his work or the family. He texts me about Alzheimer's disease, because he's worried that I'm not staying up to date, I guess, and so he keeps sending me what he learns. But I appreciate it, because I get to hear about cures for Alzheimer's disease, say once a week or so. And they cure Alzheimer's in mice on a regular basis these days. And so I get to hear about that from my dad, and of course I get to tell him each time that yes, I was aware of this study, but I appreciate his effort. But I'm going to talk to you about ways to really find the cure, the final step of how cures and effective treatments are discovered in medicine. And that is through clinical trials. This is the final proof. And as I do this, I just want to lay out the process from the beginning to the end. How are medicines developed? How can cures be found? In all modern cases, almost all modern cases, the approach that's taken is to find what causes the disease, what is the disease, what is causing the problem, and then target that in a way to change it. There was a hypothesis called the cholesterol hypothesis in the 1950s. That hypothesis proposed that too much cholesterol causes heart attacks and strokes through atherosclerosis. Bacon and sausage, like we were provided this morning, were common in the 1950s. They still survive in some breakfasts, but the cholesterol hypothesis was then proven with a class of drugs which demonstrated that lowering cholesterol, the bad forms of cholesterol, can actually prevent heart attacks and strokes. And that's been remarkably successful. Many millions of people have benefited from that. So I'm just going to take a few slides to point out what we think causes Alzheimer's disease, where are the drugs that are targeting it, and where are we in terms of the final step, proving in clinical trials whether a drug works or not. I think many of you are familiar with the effects of Alzheimer's disease on the size of the brain. The brain shrinks because of the damage that Alzheimer's disease does, the loss of neurons, the thinking cells in the brain. Because of that shrinkage, we can see that here on a brain donation autopsy. You can also see it in the imaging, the MRI scans that were just discussed. But as we zoom in on the actual brain tissue itself and try to determine why the brain is shrinking, this is what is seen. You can see here these highlighted by arrows. These are called amyloid plaques. Here's another amyloid plaque. There are also tangles in neurons. The neurons get these tangles inside of them, and these plaques are on the outside of them. It was then discovered that these plaques are made of proteins that we all make in our brains. Our neurons produce this protein as part of normal physiology, normal workings of the brain, but then something changes, and this amyloid beta builds up in these plaques to very high amounts. This is what's called the amyloid hypothesis. This has been postulated for almost 3 decades now. This amyloid hypothesis was then targeted with drugs. Drugs were designed to try to intervene in the amyloid and change whether it can do damage in Alzheimer's disease. Some of the first treatments were active immunizations that created the immune system to attack the amyloid. That was a trial in 2006 called AN1792. Later discoveries, one of which was actually at Washington University by Dr. Holtzman, found that an antibody could bind to amyloid beta and change the risk of those amyloid plaques in mice. That, I probably received a text from my dad about that, Dr. Holtzman. Other treatments target the production of amyloid beta. They try to block it. All treatments can have side effects. There could be issues in manipulating the system in this way. But the goal is to get a relatively safe treatment that can change what we believe is the cause of Alzheimer's disease and do it so that it helps our patients. This is a list of these approaches. This is what we call disease-modifying therapeutic attempts. These are trials that have been performed where people have tried to target these pathways. I mentioned the study that ended in 2006, AN1792, the active immunization. There's a list of studies here where drugs have attempted to make this change. Some of the more recent ones, you'll see the bapineuzumab and solanezumab, some of you may have participated in. Recently, these studies have ended, and they did not meet the critical threshold that's required to prove that these drugs are effective. However, one of them, solanezumab, had data that suggested it might have helped people with mild dementia, an earlier stage of dementia. And this is related to what Dr. Morris was saying at the beginning of the discussion today, that we think that treating earlier in the disease is likely to be more beneficial. And so the field is focusing on the earlier stages. And from this, several trials with solanezumab have been started, and later I'll review some of those. But this raises questions about how we run our clinical trials. This last step that is necessary to prove whether a drug is effective, how do we design it? Who do we enroll? Do we enroll people who have Alzheimer's disease already at the moderate or severe stages, at a mild stage, or even before they have any symptoms? Do we enroll people who carry very rare mutations that can cause the disease, or enroll people who have amyloid in their brain but are cognitively normal? When do we treat these people? Do we treat them years or decades before the first symptoms begin? Do we treat them once the very slightest symptoms are detectable? Or do we treat when dementia symptoms start interfering in daily life? What do we target? Do we target the amyloid that I've talked about? How do you target it with the different approaches? And then how long do you run your trial for? What do you monitor? How do you know if the drug is successful? The answer is not a specific answer to any of these questions. We don't know the answer, which is why there's still questions. So I like the approach of you take all of these approaches. You attack it on every level possible and then figure out what works and what doesn't. So in clinical trials, many of you may know what a placebo is. Placebo is oftentimes interpreted to mean nothing. It's actually not nothing, right? It's everything but the active ingredient, which sounds like nothing, right? So, but the point of a placebo is not to do nothing. It's to have a control. It's to have some way to know whether your drug is working. If we had a drug and we gave it to everybody and everybody was cured the next day, we wouldn't need a placebo. We don't have a drug like that. So because we need to know whether a drug is effective, we are required to have placebo. And everybody that I talk to, my patients, and I'm guessing most people, when they enter a clinical trial, they say, I don't want to be on the placebo. There's two things about a placebo. It's usually blinded, so I don't know, and the person doesn't know, and we have no control over this. Some computer is deciding this on a randomized basis. But in fact, I tell people, well, you may want to be on the placebo. And they say, why would I want to be on a placebo? Well, sometimes drugs don't help. They actually hurt. We don't know. That's the point of a clinical trial. It's designed to help. We hope it helps. We're trying to prove if it can help, but we don't know. And without a placebo, we'll never know. A drug will never be approved without a placebo, unless it's miraculous. So placebos are required. They're necessary. The government requires it. The FDA has it as a required rule. So I'm going to give you a very biased brief history of Alzheimer's disease. It's biased from my standpoint in our studies and what we've been doing, but I want to point out just a few things. One is that Alzheimer's disease has been with us now for all of history and probably before. The recorded documents, Chinese and Egyptian scrolls that date it at least back to 3000, 4000 BC, and probably before. So what is described as senility, the progressive loss of cognitive abilities associated with older age. We believe today that that was likely Alzheimer's disease in many cases. In 1906, Dr. Alzheimer's described the first cases of what later became known as Alzheimer's disease. He described those amyloid plaques and the tau tangles. And in the early 1990s, mutations were discovered that unveiled the molecular biology of how these can cause disease in people. And it tied together those amyloid plaques with people who actually get Alzheimer's disease. We call those dominantly inherited Alzheimer's mutations. And through the 90s and in the 2000s, the first drugs were being developed to target that particular part of it. Those antibodies I talked about against amyloid beta and small drugs which block the production of amyloid beta were being developed. And then that process of developing those drugs, testing them in animal models, bringing them forward into safety studies, testing them in people, moving through this entire process, it's a regulatory process, takes about a decade before these drugs were first used in clinical trials. And now in 2012, as Dr. Morris mentioned, we're taking these drugs and targeting people even before they have the first symptoms, what we call a prevention or a preclinical study. So you can see how the field is evolving here and how it's accelerating. We're learning quite a bit over the past few decades, and this is the hope. The hope is that we will be able to do what was done with cholesterol in the 70s and 80s. In the 50s, there was this hypothesis called the cholesterol hypothesis. In the late 70s and early 80s, people who carried mutations that caused very high cholesterol levels. You see in these photographs the backside of a young woman's ankle. She's 30 years old. She has cholesterol deposits in her skin from very, very high cholesterol levels. She was given a drug that lowers cholesterol levels in her blood, and a few months later, on the right side, those cholesterol deposits melted away. Some years after that, it was proven that that class of drugs that today we know as statins can prevent heart attacks and strokes, and takes these family members with these mutations and gives them normal lifespans where they were dying in their 30s, 40s, and 50s from heart attacks and strokes. Now they're living into their 70s and beyond. So the question is, can we do the same thing? Can we take drugs, target amyloid, and you see here one of the scans that's been shown this amyloid deposition by PET scan. And if we target this and other forms of amyloid, can we change it back towards normal? Can we normalize the amyloid beta biology, the physiology? Can we block its toxic effects on the brain? And if so, can we help with the disease? And so I'm going to now talk about a few trials that will be offered soon here at Washington University that take this approach. And as I mentioned, one of the drugs was actually developed and discovered here at Washington University by Dr. Holtzman, and that's solanezumab. And that drug was chosen by a committee who was deciding drugs to be used in this trial called A4, Anti-Amyloid Treatment in Asymptomatic Alzheimer's. And so who is this for? This is for people who have normal memory and thinking, aged 65 to 85, and who are willing to learn whether they have amyloid deposition in their own brains. As part of this study, people will receive a PET scan to measure amyloid, and they will be told whether it's elevated or not before any symptoms are there, before they have any symptoms. And this is a first. This hasn't been done as a matter of research before on a large scale. And when are we targeting? Well, the drug trial targets people who are asymptomatic, as I mentioned, no symptoms, who have the amyloid. I mentioned the drug is solanezumab. It binds the soluble forms of amyloid beta. And it will be for 3 years long, infusions every 4 weeks. There'll be other tests in it, MRI scans, safety checks, EKGs to monitor. There'll be close monitoring and cognitive tests. But the goal here is to answer the question, if someone is treated with this drug for that time duration, does it prevent cognitive loss over the 3-year time span? And if it does, this drug could be approved for that use. This is the last step in the process, what we call a phase 3 trial. There's another study. I mentioned that all approaches should be taken, not just amyloid, but other approaches, and that includes an approach to target the metabolism of the brain. There were scans shown where the metabolism in the default mode network is decreased in Alzheimer's disease, where the amyloid deposition is. Researchers have been studying the effects of insulin that's sprayed directly into the nasal passage, and that insulin gets into the brain. And it's thought that that insulin could improve the metabolism of the brain. And so it's never a good study unless you have a witty acronym. So SNF is the study of nasal insulin to fight forgetfulness. So who is this for? This is for people who have what we call mild cognitive impairment or very mild symptoms of Alzheimer's disease. A clinical dementia rating of 0.5. The age range is wider, from 55 years old up to 85 years old. Stable medical conditions, but importantly, a person cannot be taking diabetes medications because this insulin is going to be sprayed directly into the nasal cavity. It won't affect the rest of the body very much, but it will get into the brain. So 2 sprays a day, having a study partner to assist with the study, and it will go on for each individual for 18 months. Will include blood work, EKGs, MRI scans, a lumbar puncture for cerebrospinal fluid, and neurological testing. And then there's the DIAN-2 adaptive trial that Dr. Morris mentioned, which was launched at the very end of 2012. And this is for people who carry a very rare mutation in one of 3 genes. It's so rare, I would guess that probably nobody in this room has one. There are about 5,000 to 10,000 people around the world who carry these mutations. And the goal here is to treat people who either are completely normal, all the way up until mild dementia, with one of several drugs, and there's 2 drugs being used. One of them is solanezumab that I mentioned earlier, and a different drug called gantenerumab. So people will be treated with one drug or the other drug, and additional drugs may be added. And so this study starts for 2 years and may extend upwards of 6 years in total duration. So I'd like to end with, if you're interested or know someone who may be interested in these studies, how do you find out more information? There are some folks in the back for the A4 study, Pam Millsap. And Pam, can I see? There she is. In the back left corner. And if you're interested in the A4 study, no symptoms, taking a drug for 3 years to see if we can prevent cognitive decline, or know someone who might be interested, please talk to Pam. The SNF study, Joyce. There's Joyce, and to the right, good. They've geographically, strategically located themselves. So you go to the right, or maybe your left, to see Joyce for the SNF study, and that's for mild symptoms, to see if that insulin in the brain can actually help with the symptoms. And if you know of someone who has one of these very rare mutations, then you can contact Wendy, who's in the back of the room, now standing up in the middle, for the Diane 2 trial. Okay. Thank you for your time.
Why don't you just stay up here? Thanks, Randy. I'm going to ask Danielle, Jennifer, Dave, and Manu to come up and join the panel. We're running just a tad behind. I did want to tell you about the results of a study that some of you participated in, trying to find out if you would like to learn whether you have a positive amyloid scan, whether you have an abnormal tau level. We haven't been doing that. When you join the study, you get a consent form and you sign it. You agree to participate, and it says in the consent form, we're not going to tell you your individual research results. We've had some discussions about this, and some of you feel pretty strongly you should know that. So we interviewed about 220 individuals in the past couple years, and we wanted to know your attitudes about getting your individual research results. Then we split you roughly in half, 119 people. Then we're told what learning your own research results may or may not mean for you. And the 103 had a second a placebo, if you will, as Dr. Bateman mentioned. We just told you about Alzheimer's disease. We didn't tell you about the consequences of learning your research results. Some of the risks of learning your research results is many people, if they find out their amyloid level is elevated on a florbetapir scan, that's one thing, but what they want to know, or many people want to know, well, what does that mean about my risk of becoming demented? Will I develop dementia? And the answer is, we don't know. So we can't tell you what that finding means. Potentially, of course, if people learn about such things, it may make people ineligible to do certain things, including be eligible for long-term care insurance. Insurers don't want to enroll people who they think are going to need their insurance for long periods of time. So these are potential risks. So what we found out, just to look at this, 94% of the surveyed people say, yes, tell me my results. And 90% says, I actually have a right to that. That's what you said initially. Then we gave that intervention, and the placebo people, this is under the post-intervention segment of this slide, after they learned about Alzheimer's disease, their attitudes were the same. 94% still said, yes, tell me my results. The group that got the intervention about the risks, 80%, 79% still wanted to know, but there was a significant decrease once people learned their risks. So there were a couple factors. We're still trying to understand the results. How people responded varies depending on whether they think that they're at risk for Alzheimer's disease. If they're at strong risk, they still want to know their results even after learning about the potential consequences. the participants meeting. If people hadn't been to this meeting, they still wanted to know their results even after the educational piece about the risk. But if people attended more than one of these meetings, then they had a significant decrease in the people who want to know. Other factors is if you had a demented parent, how much education you have, and what actual research results do people like to learn about? Is it their test performance on the memory test, the PIB scan, the CSF levels? I won't spend a lot of time on this. We are very much governed by ethical principles. One of them is autonomy. That is, you, the research participant, should have what your decision about whether you wish to participate in research must be honored. We want to do good for you as a person as well as for society, and we want justice. We want everyone to share equally in the distribution of benefits and burdens. But these ethical principles can conflict. So, for example, Dr. Rose doing a driving study. Say she finds that a driver is not so good. They're doing unsafe behaviors. So her obligation for do good for society may be to tell that unsafe driver that you're putting society at risk by driving, and yourself as well. But the autonomy may say, well, the driver gets to make that decision. So these two things can conflict. And we may find the same thing with individual research results. Here's what we plan to do. We still, as I say, need to analyze the results further. We'll probably collect more information with an additional survey. But we're already consulting with our institutional review board, the Human Research Protection Office, that governs what we do, about returning individual results to participants. We also have to determine the effect of this on our research. So if people find out they're not at risk for Alzheimer's, they have a negative scan, maybe they'll drop out. I don't know that, but I mean, that's a potential. Or if they're convinced that they have on the road to Alzheimer's, maybe that will affect how they perform on some of the tests. So we have to factor all this in. But we listen to you. I want you to understand this. The message is clear. Many, the vast majority want to know the research results. And so after all of these steps and after we gather more information, we will consider how our current policy, not returning the results, will be changed. So I just wanted to give an update at the very early returns. Now is the time for questions and answers. So our Memory and Aging Project staff is circulating among the tables, and they're going to bring the questions up to me, and then I'll distribute them to our panelists. But while our 5 speakers are here, and Krista, just come right on up. While our 5 speakers are here, let's give each of them a round of applause for all that they did. Thank you. All right, Dr. Holtzman, what's the chance of some problem during the spinal tap?
It's a good question. It's a question we get all of the time. And a spinal tap is actually not a risky procedure. Most people tolerate it like drawing blood. But the major risk is that a small percentage of people can get a headache after the spinal tap. Usually it lasts not that long, but it can last for more than a day. The older you are, the less likely that you would get that complication. So in people over the age of 60 or so, maybe only 1 to 2 out of 100 people will have that problem. And if you do have it, we can take care of it with another procedure called a blood patch. So the bottom line is that they're very safe procedures in general.
Dr. Bateman, there was a report in the Washington University Magazine about another trial in Alzheimer's, possibly relevant to Alzheimer's disease, about antidepressants. What do you know about antidepressants?
The study that you're probably referring to, and by the way, my dad texted me on that study as well, even though I was a co-author on the paper, and he didn't know it or recognize it. So So in that study, what was shown is one particular kind of antidepressant called citalopram actually lowered down the amount of amyloid beta, which was made by both mice and people in a study, in a CSF study, a cerebrospinal fluid study. And that is a study which is supporting the original finding that people who take antidepressants may have less amyloid deposited in the brain. And the theory is if an antidepressant like citalopram can lower amyloid beta's production or increase its clearance and lower the percent of people who have amyloidosis, then could this lower the risk of Alzheimer's disease? Not just in people who have depression and need the medication for that, but maybe even in people who aren't depressed. And so I think that's the implications of the study. Probably not, but I'll try. The question was, can I spell citalopram? I feel like I'm in 5th grade in a spelling bee now. Citalopram, C-I-T-A-L-O-P-R-A-M is my best guess.
Yeah, that is correct. And what is the brand name? What is the trade name?
Is this bonus round now? So,
well, most people will know about the...
Celexa, C-E-L-E-X-A, is the brand name of citalopram. I want to caution everybody, though, that you should not go out and ask your doctor for citalopram to prevent Alzheimer's disease. Don't do that. We really don't know, and let me loop this back to the point about clinical trials. It is really, really important that a clinical trial be done to demonstrate whether that medication or other medications like it can actually help prevent Alzheimer's disease. I want to tell us another historical related story to you all about a drug that was thought to help prevent Alzheimer's disease. In women, it was estrogen. And on that basis, and on the basis of many other things, people gave estrogen to women by the millions with the belief that it was going to help. And then a very large study proved that it not only did not help, it hurt. It made the risk worse. So this is my word of caution. Don't go take drugs until the clinical trials show it's helpful.
Not only was it worse, there was an elevated risk of cancer and deep vein thrombosis. It actually hurt.
Yep.
All right, we have to move on. Dr. Morris, that's me, I guess. If you decide to give results of research to everyone, can some participants decline? Of course. Of course, it will be an individual option to do that. Dr. Goyal, I'm scheduled to get some scanning, MRIs, PET scans, maybe as many as 6 different scans. Why can't I just get that short combo of PET MRI that you talked about?
That's a good question. Different PET scans require different injections, and they can't be combined into just one scan. So that is one reason you can't do it all in one scan. And the same thing with the MRI. Different MRI scans require different scans that need to be done sequentially. Now MRI scans can all be done in one session, but unfortunately they can't all be done at the same time.
Okay, and a follow-up on that, is there any communication once the person is actually in the scanner? Is it just complete silence, or are people talking to them? What's going on?
No, absolutely. There is a trained technologist that will be there monitoring the scan continuously, and they will be able to communicate with you while you're in the scanner. In addition, you'll have access to a bulb or some type of device to be able to communicate back that you need if you were to have some type of problem during the scan.
Thank you. Danielle, this person is not supposed to get up on ladders anymore. So how do the smoke detector batteries get changed?
Good question. Well, as once mentioned, you do not get up on ladders, so if you're doing that, please don't do that. But there are several other things that could be done. There are adaptive devices that people can use to reach things that are up high. we can always have someone to help us. And even if we don't have the finances to have someone to come in and help us, that's where we come in. We can recommend different community organizations that can pull someone in the home to do things, maintenance, things like that. And once again, assistive devices, if you don't have finances for those, I can definitely give referrals for community organizations that can help with finances to get assistive devices.
Okay, thank you. Here are two questions. Are there opportunities to participate in the Knight ADRC in other places, such as Hawaii? And another person is moving to Tampa, Florida. So each winter, we will set up a new Knight ADRC site in Honolulu and Tampa. No, we can't. If we can't support our West County, we're probably not going to be able to support Honolulu. But we do all we can to continue your participation. So, for example, if you were to return from Honolulu to visit friends or family in St. Louis, or from Tampa, we would work with you to coordinate your assessment when you came back. And if you have granted us provisional consent for autopsy, for which we're very grateful, we can work with local people in Honolulu, Tampa, Milwaukee, wherever you are, so that your wish can be granted. In other words, if you have given consent for your brain autopsy, that still continues, and we will still continue as our participant and hope to see you again. Now, Dr. Goyal, someone wants to know about open MRI. What is that? Do we have it? And what's its utility?
An open MRI is designed to relieve some people who experience claustrophobia in a scanner. Yeah, it certainly is real. The problem with open MRIs is that they're not strong enough to be able to see the details that we need to see in the brain. And over time, our MRIs have become larger, but that doesn't completely get rid of the claustrophobia, and that is truly a real problem with the MRI scanning that we have to deal with.
Here's a question. Someone says, I'm over 85. Do you want me to participate? Absolutely. In some of the studies that Dr. Bateman mentioned, particularly the A4, the study designers for that trial said only 65 to 85. That wasn't our decision. But in the Memory and Aging Project, remember, what was it, 22 to 103 or something like that? We certainly will have you in the Memory and Aging Project. I have a couple of brothers who would be eligible. Do we enroll siblings? Absolutely. Siblings are more than welcome. Dr. Holtzman, just how do you keep a mouse awake for 4 weeks?
Well, that's a great question, and one that we took a little bit to figure out how to do that. It turns out that people have been studying sleep in rodents for some time, and one of the techniques that they came up with that we employed was it's called the flower pot technique, where you basically take a mouse and you put it on a very large platform over water, and in that case the mouse is fine. It can go to sleep, be awake. But if you put it on a very small platform that it only has enough space for the mouse to sit on, but no more space, if it falls asleep, it would fall into the water. So it makes the animal stay awake. And so there's other techniques as well, but that's the one that we utilized. So just feel fortunate that you're not a mouse. So.
Okay, Dr. Bateman, I think you said that statins were developed. You gave that as a historical precedent and have really been remarkable in reducing cholesterol and atherosclerosis. But can't statins cause memory problems?
So, my dad's going to kill me, but he texted me that as well. So no, it's commonly reported in the literature that statins, in the popular lay press and newspapers and things like that, that statins can cause cognitive problems. And there's been some research into this area, and it's actually very difficult to demonstrate that statins themselves cause memory problems. And what that probably means is that statins, by far and away, probably don't cause memory problems. But having risk factors and diseases and illnesses that a person might be on statins for, high cholesterol, heart disease, strokes, those things may be associated with memory loss. So to my knowledge, I'm not aware of a clearly definitive study that has any relationship between statins and memory loss.
Okay, Jennifer, all the studies we talked about today really focused on people who still were asymptomatic or cognitively normal and very mild Alzheimer's disease. What about care for a person who is a little more progressed with Alzheimer's disease, dementia, and particularly for their caregivers? What would you recommend?
For individuals who are experiencing the changes that are inherent to the progression of the disease, we're very fortunate that we have an extremely strong chapter of the Alzheimer's Association here in St. Louis, and they offer a whole variety of services, support, consultations, resources, and referrals to really see people through the entire spectrum of this disease. There are some representatives from that association here. I know Stacy is in the back, the chapter president. And certainly you're always encouraged to reach out to them at any time. They have 1-800 numbers that you can call 24 hours a day, 7 days a week. And if you are interested in reaching out to them, their local numbers 314-432-3422. And you can contact them anytime or check out their website at www.alz.org. And if there are any questions that are not answered there, we certainly encourage you to check out some materials we have in the back. Ms. Murda Spencer is holding up an information brochure right now, and you're welcome to access those, and they will get you set on the path to what you need.
I indicated earlier that we have two of the best social workers anywhere, Terry Hostow and Becky Fearberg. And for all of our Memory and Aging Project participants, if there are those needs, Becky and Terry can certainly help indicate resources, most prominently our St. Louis chapter of the Alzheimer's Association that can help. So we try to be covering not only research, but to direct people to the areas where they can get support. So one of you called us on something, and that's appropriate, and that is we tend to use terms and abbreviations that are familiar to us but may not be familiar to you. So one question, once again, is where does our grant funding come from? I think I mentioned, but I'll just reiterate, that it comes primarily from the National Institutes of Health. And of the institutes, primarily the National Institute on Aging, although we do receive funding, grant support from other institutes at the NIH, National Institutes of Health. We're also supported in other ways. The Alzheimer Association, the National Alzheimer Association, gave our Diane study and our Diane Trials Unit study a very large gift. We also get foundation support, and we also are very well supported by our participants and our friends. So many of you not only give your time, but you also provide us with research support. This is not a call for you to give money, but I will just point out that I told you that we just reapplied to the National Institutes on Aging for research support for another 5 years for our Alzheimer's Disease Research Center grant. We began planning the research that went into that application last summer. And if we're fortunate enough to be refunded, we'll get funded next May. So that's a period of almost 2 years before we know we'd have the funding to do the research. Things will change. If we get funding from foundations and from philanthropy, that allows us to fund exciting new research immediately without this long wait. Now, in terms of abbreviations, we use things such as MRI and PET. And someone wants to know, does that mean that it's a household pet? But maybe, Manu, you can explain what those abbreviations stand for.
My pleasure. MRI stands for magnetic resonance imaging, and it represents a way to use a very powerful magnet to image what's going on in the brain. PET stands for positron emission tomography, and those are concepts from physics that allow us again to image the brain with an imaging tool.
All right, a couple of things. Thank you very much. First, there was a question about frontal temporal dementia, and maybe somebody has been diagnosed with that. Are we studying that in the Memory and Aging Project of the Alzheimer's Disease Research Center? Dr. Ghoshal, will you stand up again, please? Please see Dr. Ghoshal. That's her particular area. Wave so everyone can see you. Keep waving, keep waving. If you have frontal temporal dementia questions, please see Dr. Ghoshal after this. Second, it is 11 o'clock. We're supposed to end. I think we would be happy to stay. If you have to go, please go. I'm told that shuttles will pick people up at the front door to take them to their car. Pretty nice. So we'll try to get through a few more of these. Here's a question about whether if people sign up for those clinical trials that Dr. Bateman discussed, the A4 study or the SNF, what does that do for their participation in the Memory and Aging Project? Well, we will continue to see people, but it will, particularly the A4 study, will probably change things for some time. That's a lot of, what is it, an infusion every 4 weeks for 3 years, I think, right? So we have to monitor burden on the participant. But remember what I said, whether you go to Honolulu or Tampa, you're still part of the Memory and Aging Project, and we will work with you to try to continue to do that. There are lots of questions, Dr. Holtzman, about sleep apnea and CPAP and effect on Alzheimer's disease. What about that? First of all, if you're already diagnosed with sleep apnea, are you eligible for your sleep study? And second, what about this idea of the sleep-wake cycle, this diurnal rhythm? Is that affected in Alzheimer's disease?
Let's first talk about this obstructive sleep apnea, which is one of the most common disorders of sleep. I think it is the most common. I think in people over 60, it's as many as 25% of people can have some form of sleep apnea. And if you, at the end, if you want to ask Jennifer some more questions about this, she's an expert, but
Jennifer, right next to you?
No, not this Jennifer, but Jennifer McClellan, who's in the back. And I think we don't know whether treatment of obstructive sleep apnea with a method called CPAP, or continuous positive airway pressure, will help prevent Alzheimer's disease. We know it's a very good treatment for sleep apnea, and there are, one of our investigators, Yoel Ju, is investigating the effects of CPAP treatment in people with obstructive sleep apnea to see if it might decrease some of the amyloid that's being produced in the brain. So we're actively investigating that. In the study that we're starting in the Memory and Aging Project on sleep, when people enroll in the study, we aren't taking people who know they have sleep apnea because that's an exclusion. However, if they happen to have it and they've already entered, we will continue to follow them during that period.
And one other related question, I'm taking Ambien to help me sleep. One, does that increase or decrease my risk for Alzheimer's? And two, what about eligibility for the study?
That sounds like a question that Dr. Bateman's father might have texted him, but I think we don't know whether Ambien is protective. We're studying a similar drug. Actually, Brendan Lucy, who works with Dr. Bateman, is determining whether drugs like Ambien might lower amyloid beta. And the second question was whether
Eligible? If I'm on Ambien, am I eligible to participate?
not for the study that we're carrying out, but some of the studies that Dr. Bateman and Lucy are carrying out that might be possible.
And maybe, John, can I add to that?
Sure.
So if folks are interested in signing up for sleep studies, where some of you may remember that in my lab, we presented several times a study that we did with 100 of you enrolled to have a CSF catheter placed. And I know some of you folks are here. I saw Bonnie. Could everybody raise their hands that's done that study, had a catheter and had CSF taken over 36 hours? A few people. All right, I thought, yeah, okay, all right, so maybe about 10 or so. So in this study, there's a catheter and the CSF is collected once an hour. So there's one spinal tap. We collect the CSF over that time, and it's a sleep study. And what Dr. Lucy is doing is there are three groups of people. One is sleep deprived. You'll be kept up by a nice nurse. You won't be put on a platform over water. The second group is just normal sleep. You sleep like you normally do. And the third group is actually given a drug somewhat related to Ambien that causes people to go into a deep sleep. And Dr. Lucy thinks that may change temporarily the amyloid beta levels, and if that can be demonstrated, it will lead to other studies. So if you're interested in that, you can go to the back, and Wendy Sigurdsson, yeah, back there waving, has that. And we're also resuming the blood test study. So 60 from the original 100 and additional people, if you're interested in doing this, we'll be working on detecting blood amyloid beta in these kinds of studies. So just see Wendy and Kara, who's the medical student here, about those studies.
So thank you for mentioning blood tests, because there were a number of questions about that. So I'm just asking, so we're telling people to see Jennifer and Becky and Terry and Wendy and Kara. Where are these people? Are they outside, or where are they? Well, yeah, so we need to have all of our coordinators in a central space so people can find you. So there's going to be meeting outside. Okay. So we think, Howard, can you verify that those, can you just let me know now? Or Mary, thank you. Is that where they are? Okay. So as you leave, the people for sleep and for blood test and for other things will be outside for you. All right. So Danielle, is there a way to get a copy of this home safety checklist?
So the particular home safety checklist that we will be using as a part of MAPS, you have to purchase the informational guide for, but there are a number of different home safety checklists that are available online. You can, if you are a person that uses the computer, I always tell people it doesn't sound very professional, but if you use Google or different types of ask.com, management type websites, you can definitely look for home safety checklists. And then also your local libraries, if you could ask your librarian, they can also help you find some online. Those are the ones that I typically recommend for people that are looking for them.
Alzheimer's Center has it too.
Oh, Alzheimer's centers?
What's an Alzheimer's center?
The Alzheimer's Association has.
The Alzheimer's Association.
The Alzheimer's Association. So,
Okay.
yes.
Thank you. So a number of questions about things that might decrease risk for Alzheimer's. Coffee, green tea. Does it decrease the risk for Alzheimer's? Alcohol. How many have heard that red wine may protect? By the way, we talked earlier about our offerings for your breakfast, including bacon and sausage. But you know what's changed now? Dr. Holtzman says he goes around the world, and I can tell you we have lots of these breakfast meetings, and they used to be not very heart healthy, but increasingly there's fruit. So did you notice we had fruit? How many of you had blueberries today? There you go. Maybe next year red wine? What do you think? Should we put... So unfortunately, none of these things have been shown to cause a decreased risk of having Alzheimer's disease, to cause a decreased risk. Let's take alcohol. By the way, I don't know if you know this, the first report saying that drinking red wine decreases your risk of getting Alzheimer's came from a group in Bordeaux, France. So a conflict of interest, right? So let's think about it. Maybe it does. Maybe it does decrease your risk. There are lots of things people have studied. Some of the things, including resveratrol. Who's heard of resveratrol? That's in the skins of the grapes. And maybe that's protecting against Alzheimer's disease. Maybe. But no one knows directly. It may be the people who are accustomed to having a glass of red wine in the evening are following other dietary practices or have a different set of maybe they can afford to have a glass of red wine every dinner. Maybe they're well off. And maybe it's those things that are protective. It's not the red wine per se. Does that make sense? We don't know yet whether it can help or not. Someone wanted to know about whether anesthesia can increase the risk. There's a lot of interest in that, and I have to say, just as Dr. Bateman was discussing mixed results about some studies such as estrogen, and people thought it might protect, but when we really studied it, it made things worse. Well, the story isn't clear on anesthesia. Some people think that it does increase risk of memory decline after the anesthesia. Other people, including us, have done some studies in you. We ask you every year to update your health history, and if you've undergone a surgical procedure in which anesthesia was used, we look at your test performance over a long period of time. And guess what? Those of you who had anesthesia and surgery didn't decline any differently than people who didn't. So this group, at least, it doesn't seem to affect it. But these are all very important issues to still determine. Here's a question, Randy. I think it's important if someone's in the A4 study, since there are so many infusions every 4 weeks, every month, does that mean they can't go on vacation anymore? Would they have to stay in town to get the infusions? Can they go somewhere else?
You can take
Hawaii?
a 3-week, 6-day vacation anywhere you like. No, I think right now the center where we will do the infusions will be in St. Louis. If someone permanently moves, there may be the potential for a transfer, although that's not recommended. But I think as far as we're planning now, it will be St. Louis where we'll be providing the infusions. So someone should really plan, if you sign up for these kinds of studies, you want to plan on being in St. Louis for the majority of the time. Short vacations are okay if you go somewhere for 3 months or so. As long as you can get back for your infusions, that's okay as well.
Jennifer, the DUO program in part is to help decrease stigmatizing Alzheimer's disease. What does that mean for the individual? Do you think that people are so concerned about having that label that they deny that they have the illness?
I think we still see quite a bit of that throughout the variety of people who have Alzheimer's disease. Granted, that's a little bit less in the DUO program because the individuals self-select to be involved, and so they're obviously forthcoming about the diagnosis. But this is something that the students even dealt with. For example, one of the DUO participants was out at an event with his student, and they met, a random acquaintance, and the acquaintance said, well, how do you two know each other? Because it's a little unusual for a medical student to be hanging around, with somebody who's not their grandparent typically in that age situation. And so that became that moment of wondering, do I say something? Do his friends know? What do we say? And so there is still that stigma that's out there, and knowing what people are comfortable sharing and what they're not. And so that was a great example for this particular student of how somebody with these changes related to Alzheimer's disease addresses that with their acquaintances and what that actual situation looks like. I think through little interactions like that, the opportunities present themselves for both participants in the duo match.
All right. There was a question about brain autopsy and whether the autopsy does look at plaques and tangles. So the answer is yes. It's not limited to that. It will look for any abnormality in the brain tissue. And a full report will be generated and sent to the family, the survivors. So the family will know what the brain autopsy results showed. Now it's 11:15. I'm going to say that we'll pull this to a close, but I think our speakers will be able, hopefully, to remain. And if you have specific questions for them, you had so many good questions, we simply could not get through them all. We'll try to keep them all and see if we can write up some in our newsletter. But will you, thank yourselves for coming today, and